Yeah, so first, our primary focus will try to answer the three different directions. The first one is the calcium image, because we know that calcium is very important for our cellular cell function and also our whole human body. So we try to answer and to understand how these small calcium work in our cells. And then the second part is they will try to understand the organelle communication. Because we know that there are a lot of organelles in the cell, and how these cells can interact and communicate with each other is also our focus. The third part is that cancer biology. Actually, a lot of function can apply and answer the clinical problem of cancer. So we try to use this technique, try to improve some therapeutic strategy and clinical strategy. Yeah, so first I think most of you can understand this cartoon picture is the structure of our animal cell. The biggest one is a nucleus, and which surrounded is the ER lumen, which is the major calcium storage. And then here is the Golgi, which is also the important important organelle to communicate the intracellular signaling. And the other important one is the mitochondria, which is the power supply of our cell. And what I mentioned earlier is that all of them is very important for the calcium signal. So first I want to say that if the organelle or every structure with the membrane cover, they have their calcium channel. So how these calcium work for our body, that is very important for the muscle contraction, neurotransduction, and cell death, also cell proliferation and the gene transcription. So if we can regulate the calcium signal very well, maybe we can regulate these important signal. Yeah, so how we try to control the calcium? We generate a system called optogenetic. So we merge the protein with the calcium channel and also some important channel in the organelles. So when we try to control these light-sensitive protein, which can help us to control the calcium channel open and close. So here is a picture that we try to apply this technique to see how calcium oscillation in the cell. So when we turn on the lights, we will increase the calcium influx and turn off the calcium efflux. So you see a lot of peak here. And to show the peak, to show the video here, I think which can help you more understand what we are doing. So you can see that here is like a peak here. But actually I have another two, this one. So we can compare the white that means the mitochondria. So if we apply the calcium-related chemical, it can regulate the movement of the mitochondria like this and this. Yeah, and through our microscope technique, we can record and analyze how these mitochondria move. Okay, so the second part is the organelle communication. Like what I mentioned before, the major character which can think extracellular and intracellular signal is the Golgi apparatus. So now we try to understand how Golgi works, especially in the metastasis. And now we find that the Golgi stress is a very important, very important signal to induce cell to have the metastasis ability. Yeah, so here is our project now. So we really hope someone can join us to answer this question. And so the last part is the cancer biology. So we know that why this cancer can induce is still a big question have unsolved now. So we only know that the carcinogenesis will induce the normal cell to have a lot of mutation, and this mutated cell will out of control to growth a lot of out of control proliferation, so become the cancer. And now we try to understand how environmental carcinogen works for carcinogenesis, like the cooking oil form and how they induce the lung cancer. And the second part is that we want to know that how these cancer overcome the stress of the chemical like chemoresistance, and what's the underlying mechanism in the chemoresistance our focus project now. So just a brief introduction. We are the group of researchers. If you are interested in the chaos image, we are very welcome. You can join us.